Thompson. I did my project on my dog's ear biome. He has chronic ear infections, and so I saw this as a perfect opportunity to figure out why his antibiotics are not working. So what I did was I first took a sample from his ear. I withheld his antibiotics for 2 weeks, then I took a sample of his ear, planted it on meal-hidden agar, and watched for growth at 37 degrees. It did not grow very well at first, so I switched to TSA agar, which I found to be much better growth. Once I had a pure colony, we set the DNA that we extracted out to an outside lab that had them sequence the DNA. And once that came back, we used the RON server to edit this DNA. First, we did the triminatics, which kind of took off any crappy DNA, and then we ran through the Proca report, which put together the contigs. And once we had all of this completed, I then did a blast report on my 16S RNA, ribosome RNA gene. Once I identified that gene, I ran the blast, and with the blast, it identified it as Bacillus saphenous. Bacillus saphenous was first isolated from a clean room in Florida off from space junk that they found, and also some spacecraft. It was first found on the Mars Odyssey, which is depicted right here. And of course, I collected my sample from my dog Diesel, who was depicted right there. So to figure out if there was any antibiotic resistance in this bacterium, I ran the Kirby-Bauer test. And what that did was I planted my microbe on TSA agar, and then put a little disc that was impregnated with different antibiotics. Incubated this at 37 degrees for 24 hours, and I came back the next day to check for any resistance or susceptibility by measuring the zone of clearance around each disc. And I found that all of my bacteria was susceptible to the antibiotics which I tested, which was great for my dog, but unfortunately it was not so great for me, because I was thinking this is why he was not healing from his ear infections. So kind of threw him for a little loop there. But something interesting I did find in using the DNA and the RON was I found hydrogen peroxide resistant genes in there, which is interesting because I use a special cleaner in his ears every day, along with his antibiotics only twice a week. And that has hydrogen peroxide in it, so I'm thinking that maybe this is why it's resisting so much. Then, with some further research, I found that this microbe is also found in the environment. And I'm thinking that this is more of a transient bacterium that he has just picked up in his ears, and that is why they're always in there. Any questions?
Good, thank you.